lecture notes on Chapter 6, Metabolism and Cellular Energy for both plants and animals. As we switch gears from cell biology to metabolism, we shouldn't forget about the lessons we learned from studying the chemistry of life in Unit 1. Metabolism is a collective term. We use it, and it can mean the breakdown or the buildup of different macromolecules. Can you remember the 4 macromolecules from Unit 1? We can also use these terms for tissues and in some cases the change of free energy. For example, you consume food, but the energy stored in that meal you just ate is being converted into a more usable form of energy called ATP. We are going to look at this form of energy conversion extensively here in Unit 2. ATP is considered the universal energy form for all living things. I'm going to walk you through the lecture notes, but I'm also going to include some fun animations for you here in Canvas. These are going to be required viewing, and your mastery of these different topics will be evaluated at the end of this unit. Your exam due date is already published in your class schedule. We will compare the energy needs of consumers. These are animals that consume food, and compare their methods for metabolism to that of a producer. Producers are plants, algae, and sometimes protists that are capable of photosynthesis. Animals require energy by eating, and plants provide energy to consumers by converting the sun's energy into plant matter. We will have to use our knowledge of cellular structures, which we learned about in Unit 1. All cell types will capture and use energy, which we can refer to as catabolic and anabolic pathways. Let's take a look at our eukaryotic cell types and compare a consumer to a producer at the cell level. The only way to convert energy is to be inside the cell where special protein enzymes are at work running very specific types of catabolic or anabolic reactions. Energy is defined as the ability to do work, and work is calculated by the formula force times distance, so F times D. For consumer cells, here on the right, the process of glycolysis is a major way of converting food into cellular energy. In a plant cell, over here on the right, plant cell, the photosynthetic pigments capture sunlight and convert it into ATP. When the plant is in the dark is when that ATP energy is converted into sugars and starch that make up the overall plant structures. An easy way to remember the 2 reactions that make up the collective term we call metabolism is to think of anabolic reactions as those that build up structures, like anabolic steroids make you have big muscles in the gym, and the catabolic reactions break down everything, like a catastrophe. And if we didn't take a look already at the structure of ATP while we were in Unit 1, then let's go ahead and do that now. ATP belongs to the macromolecule group called nucleic acids, and it's made up of 1 adenine, highlighted here in green, and it's covalently bonded to a 5-carbon sugar called ribose. And off to the right here is a series of phosphate groups linked together, forming an arm-like extension. These triple phosphate bonds are potential energy. When these are broken, the stored energy in the bonds gets released, and the cell can then do its work. Cells are involved in 3 specific categories of work. Cellular work includes transporting molecules or products to different places within the cell. Mechanical work, usually in the form of movement or locomotion, and chemical reactions. Some of these require an input of energies. The laws of thermodynamics tell us that energy is never lost. It simply changes form. Because of evolution over time, metabolic complexity varies from organism to organism. Simple prokaryotic bacteria have ribosomes, and that's all. They lack mitochondria and many of the other organelles that we would see in a eukaryote. Plants are complex in the sense that they have one set of reactions during the day and switch to a different energy converting pathway at night. Animals are even more complex and capable of switching between mechanisms that require oxygen and another pathway that allows for ATP energy when there's a lack of oxygen, called fermentation. This is what makes our muscles sore after a hard workout. the lactic acid builds up and it makes your muscles burn and feel tired. Despite the differences between organisms and the complexity of metabolism itself, researchers have found that all branches of life share some of the same metabolic pathways, and this suggests a common ancestor. Anaerobic organisms were better suited for the extreme conditions found here on early Earth. As the Earth's mantle cooled, photosynthetic organisms could take hold and eventually contribute to Earth's oxygen-filled atmosphere. So when we look inside of an animal cell, we will consider all of the functions being carried out by the organelles. One thing you can't see is the protein enzymes floating around within the cytosol. Remember, enzymes are catalysts for very specific chemical reactions and will ultimately control cellular metabolism. This is why blood glucose from a heavy meal has to be delivered to the cells. The cells must then be able to absorb the glucose and bring it inside into the ICF compartment, where it will start the process of glycolysis. Glycolysis is one of the most important metabolic pathways we'll ever study in biology. Without plants, most animals would not have any food. When we consume carbohydrates, our cells will run the catabolic breakdown of 5 and 6 carbon sugars. The glucose burn is ultimately driven by the oxygen we breathe in order to gain lots of ATP energy. The complexities of the 10-step enzymatically controlled process of glycolysis will be examined in more detail later. But just know, for now, there is no loss of the carbon skeleton. It's simply being rearranged. Here we can summarize that the 6-carbon glucose is split into 2 3-carbon molecules of pyruvate, also called pyruvic acid. This entire 10-step process that yields pyruvate only makes available 2 ATPs to the cell for it to do its work. So you might be wondering, why all this effort for only 2 ATPs? That would be a valid question. The important thing to appreciate here when we're talking about glycolysis is, 1, glycolysis can run with or without oxygen. This means when you're running a marathon and become oxygen depleted, your body can still make some ATP energy to keep you going. And sugar is one of the preferred energy sources. This is considered quick energy. And this is pretty universal among all organisms, that they can run glycolysis or fermentation. When we consider now a plant cell, the carbohydrate metabolism is an uphill anabolic process where tiny CO2 and water molecules are combined with the energy of the sun to make large 5 and 6 carbon sugars, and in some cases, complex starch. We will spend a week on plant biology coming up later in this unit. What makes a plant cell so special is the green chlorophyll. This molecule helps capture the sunlight and energize the plant's daytime metabolism. Notice the chemical equation at the bottom of this page. It runs in the reverse of the animal cell reaction. So you start out with small molecules like CO2 and water, add sunlight, and you get a larger, complex carbon structure in an anabolic or uphill reaction where you have a 6-carbon sugar, and the other benefit is we get oxygen from this process. So one of the things we need to keep in mind as we progress into the mechanisms of energy production at the cellular level is we have to consider the difference between potential energy and kinetic energy. This principle is illustrated here by the bicycle on the hill with potential energy at the top, and then the bike rolling down the hill with kinetic energy. We will be introduced to various types of energy. There's chemical energy in the form of covalent bonds, like you can see here, highlighted with the red arrows. There's also electromagnetic energy, like what we see with our eyes, and other sources like heat, sound energy, electrical, and atomic energy with the vibrational movement of electrons flying around the nucleus of an atom. The laws of thermodynamics must be obeyed, and the first law states that matter is never destroyed nor created. It simply changes form. The second law tells us that a system is not 100% efficient, and some energy will be lost as heat. Heat is not a form of energy that's available to do work. The third law states that the organization of any system has a tendency to move into chaos or disorder. Entropy is a measure of increasing randomness in the universe. So the larger the number is for entropy, the more chaos and disorder there is. Gibbs free energy describes the energy change within a chemical system. These graphs indicate an exergonic reaction where heat is released out into the universe and the reaction progresses from reactants that have a higher potential energy than the products. These types of reactions carry a negative sign. Compare that to over here in panel B. An endergonic reaction is described by a positive sign, and the reactants here are in a lower energy state than the products. Therefore, energy is drawn in from the environment to run this reaction. And like a hill that we saw before when we were reminded of potential energy, many chemical reactions require some activation energy in order to get them started. If there's a huge hill to conquer, then the reaction will proceed very slowly or maybe not at all. Protein enzymes help flatten out the hill and push reactions in the forward direction faster than what would occur without enzymes. This is referred to as lowering the activation energy. Remember, enzymes are like a deadbolt lock, and the substrate acts like a key that fits exactly into the active site, also referred to as the binding site on the enzyme. Like a puzzle, the wrong shaped substrate just won't fit into the active site on an enzyme. Once these two pieces bind or interact, they form what is called the enzyme-substrate complex. The highly specific interaction between our lock and key system results in faster biological reactions and the production of a much-needed product. The product is likely something needed to sustain life. Notice the products leaving the enzyme all the way to the right in this diagram. The substrate gets used up or consumed in this reaction. Our substrate, in green, is in limited supply because it gets converted into product. But the enzyme is still intact and ready to go another round once it finds more substrate to bind with. So the enzyme is recyclable. Metabolism can be regulated through the use of enzymes. Enzymes will catalyze specific biological reactions, and this allows for a point of regulation and control. Overproduction of products would be a waste of energy, and too little could mean there's a shortage of something that's vital for survival. Enzymes create a point of regulatory control for vital compounds. This is a negative control mechanism, which is based on feedback, is one way to control the production. And that means that when there's enough product, the enzyme pathway should shut down to avoid overproduction. There is a competitive and a non-competitive means for control over enzymes, and it has to do with that binding site that the substrate uses when it interacts to form a bond with the enzyme. Since all enzymes are proteins, and proteins need to maintain their shape, or conformation, in order to be functional, things like temperature extremes and pH can all alter the shape of these proteins and make them useless. So let's look more closely at what a negative feedback loop is. This pathway, we'll use temperature as an example. This is a tightly regulated variable. Most people can be measured for their temperature, and it's going to be around 98.6 degrees for everyone. Any change or alteration to your internal body temperature is going to set off a series of responses that will work in the opposite direction or negatively affect the disturbance. So this guy is out here for a run in the desert and is probably very hot, so his body will do the opposite and try to cool him down. One way to do this is through sweat production, which cools the surface of the skin when air moves across it. This is cooling by convection. In order to for this to work, right, we'll need a sensor to sense the heat and tell the integration center, usually the brain, that receives the information and processes it that the correct response is going to be issued, which in this case would be to trigger the sweat glands to open up and secrete sweat to cool down the body. Now let's go back and look at the enzyme itself and see how this can be a point of regulation. Metabolic pathways are controlled at the enzymatic level, and to do this, we have what are called inhibitors. These molecules can block or inhibit enzyme activity, and this works in two ways. The first is a non-competitive inhibitor, shown here in the middle, will bind in a different site from where the substrate will bind, and when this happens, the enzyme changes shape and is now unable to bind its correct substrate. In the situation of competitive inhibition, the inhibitor blocks the binding site, and the substrate can no longer bind or fit in where it's supposed to. In a classic negative feedback inhibition pathway, here we can see how the yellow product at the end of this three-step process acts as an inhibitor to the next round of enzymatically controlled reactions. This graph from figure 6.17 shows how the reaction rates are clearly lowered by the presence of inhibitors. Non-competitive or allosteric inhibitors have the most drastic effect on the lowering enzyme activity. This is a good way to control enzymatic rates. Rate is on the y-axis, and concentration of the limiting substance called the substrate is on the x-axis. We've already spent some time on the importance of ATP as our universal energy source. Now let's consider how this universal energy source is used to couple cellular reactions. In class, we discussed the energy stored in these triple bonds. Each of these release about 7.2 kilocalories into the environment in order for the cell to do work, and we discussed already the three forms of cellular work, defined as chemical, transport, and mechanical. So all of these jobs are driven by the abundance of ATP and the potential energy stored in this triple bond with this series of phosphate groups that form this triphosphate arm connected to our ribose sugar in the middle. In the mitochondria found inside of eukaryotic cells, this is the ATP generating organelle and is often referred to as the energy powerhouse. Animals will feed in glucose as the first step in generating ATP. The products from glucose include 2 ATPs and 2 pyruvates. The ATP is available for immediate energy demands, and the pyruvate molecules feed into the next phase called the Krebs cycle, where electron carriers are generated for the final step, often called aerobic respiration. This is a process of oxidative phosphorylation where 36 ATPs are made for every 1 glucose molecule. This is the payoff phase. We will compare this process to what plants are doing during the day and nighttime cycles. One very important use of cellular ATP energy is to control the ICF environment of the cell. This is accomplished by pumps that use ATP to drive sodium out of the cell and bring potassium into the cell with a 3:2 ratio. This pump mechanism is what helps maintain homeostasis for the cell. Over time, these pumps create a net negative charge inside the cell. 3 sodiums are lost and 2 potassium are brought in. Both of these are positively charged ions, but with a 3:2 ratio, you have an imbalance, and this causes the ICF compartment to become more negative compared to the outside of the cell. Because of this cellular activity and the semi-permeable nature of the membrane, cells are like little capacitors because they can store a charge. Let's do some practice. Let's complete this class activity according to the instructions. So you'll need paper and maybe some colored pencils to complete this. You are to place the 4 listed solutes in their proper cellular compartment according to their concentration given over here in the table. Once you do that, the direction of concentration gradient should be indicated by drawing an arrow for each element on the list in the direction of passive ion flow. You can email me your answers, snap a picture of your diagram with your numbers legible, and send it to me for some points. Okay, check in with you next time. Thank you.